Hello, I will talk about cholesterol metabolism today, including bile acid. So I will start with the structure and function of cholesterol, the production of cholesterol and cholesterol esters, source and fates of the cholesterol, the synthetic pathway together with its regulation, and then I will finish with the synthesis and function of the bile acids and salts. As usual, the Word file is available to you in the Basic Metabolic Pathways folder in Entrada if you wish to read this material. I also found a couple of videos, very short videos, that talk about a couple of steps which are relevant to either cholesterol or bile synthesis and utilization in the human body. And the resource material is the Lippincott book, if you wish to see some beautiful pictures and read some good words. So cholesterol synthesis is not labeled in the basic metabolic pathways diagram. The reason for that is that cholesterol is not synthesized to be stored away or to be utilized later for energy. However, cholesterol is synthesized because it has several biological and biochemical functions in the human body. Please remember this figure. You may recall that we went through glycolysis and under aerobic conditions, we produced acetyl coenzyme A in the mitochondrium, which then was used to produce energy through the Krebs cycle and terminal oxidation. Or when a lot of glucose is coming into the liver in the well-fed state, acetyl coenzyme A can be used for fatty acid synthesis and the formation of triacyl glycerides, which can be then shared with other organs for use or storage. So this is the step exactly when a lot of glucose is coming to the liver by the signal of insulin, acetyl coenzyme A is formed, and this acetyl coenzyme A can also be used to synthesize cholesterol. So although you see a lot of arrows coming from amino acids and coming from fatty acids to produce acetyl coenzyme A, under the signal of insulin, when cholesterol will be synthesized, only glucose will be used for cholesterol synthesis. The cholesterol structure is a four-ring aromatic ring structure, which is pretty expensive to synthesize, and we do not have any enzymes to cleave it. Now, what is typical of this structure? So we have four rings, a four-ring system. We have one single hydroxyl group and an aliphatic carbon side chain. So this particular cholesterol is very much hydrophobic. The only tiny little bit of polarity is given to cholesterol by this hydroxyl group. So this is the free cholesterol, which is because of the presence of hydroxy. The hydroxyl group is amphipatic. However, when this hydroxyl group is esterified with fatty acids, which frequently happens, we produce cholesterol or cholesterol esters, which are completely apolar. Now you remember that when we have lipid kind of molecules, apolar molecules, and we want them to be delivered in the blood to target cells, we need to package these hydrophobic molecules. Some of the hydrophobic molecules can bind to albumin. However, the lipid-based molecules are packaged in lipoproteins. Please remember that you learned about lipoproteins in the vital fluids block, and I put this information here just to kind of remind you what you learned about. So the lipoprotein particles are hydrophobic lipid-based molecules packaged in a single layer of phospholipids. Why do we need the phospholipids? Because the phospholipids are showing their head, which is polar, toward the aqueous environment. However, the fatty acid residues of phospholipids are facing the inside of the lipoprotein particle, so they can accommodate hydrophobic molecules, interact with hydrophobic molecules. All right, so we have a couple of different kinds of lipoproteins. If you remember, chylomicron is the lipoprotein which is put together in the mucosal cells, and chylomicron surrounds every hydrophobic lipid-based molecules that are coming from the diet. VLDL is a lipoprotein particle which is produced by the liver, and it encapsulates all the lipid-based hydrophobic molecules that are synthesized in the liver. So cholesterol, when it comes from dietary sources, will be packaged in chylomicrons, and when it is de novo synthesized in the liver, it will be packaged in VLDL. Now, you may recall also that when these particles go out to the circulation and they release triglycerides or they release cholesterol, they will become so-called remnants. So chylomicron will become chylomicron remnant and VLDL will become LDL. So please remember, hydrophobic inside and hydrophilic outside. And there are apoproteins on the surface for receptor recognition to be able to release the content or taken up by certain cells and tissues. Now, where is cholesterol? So cholesterol, being amphipathic, it is encapsulated in the single membrane. The tiny little hydroxyl group is facing outside, and the ring system is facing inside. You may recall that cholesterol is a natural ingredient of every membrane structure, and it gives the membranes rigidity. So the more cholesterol is encapsulated between the phospholipid particles, the more rigid the membrane will be. Now, cholesterol ester, which is completely hydrophobic, will be in the inner core of the lipoprotein particles. Again, just a reminder, chylomicron, VLDL, then chylomicron will produce chylomicron remnant, VLDL will produce LDL, and we have this HDL, which is actually produced by the intestinal mucosal cells and the liver almost as an empty shell, which goes out to the circulation and picks up cholesterol. So this is our savior from too much cholesterol deposition in the periphery, if you may recall. So chylomicron remnant, VLDL, LDL. All right, so let's go back to cholesterol. What are the functions of cholesterol? The function is, as I said, is a membrane component. Both plasma membrane and intracellular membranes always contain cholesterol, and this is free cholesterol, not the esterified one. Cholesterol is a precursor for bile acids and bile salts. You may recall that we talked about it in the digestive tract, that for the digestion of lipid particles, bile acids are very important to surround them and also bind the enzymes that are specifically digesting triglycerides or cholesterol esters so that mucosal cells can absorb these lipid molecules. Cholesterol is also a precursor for steroid hormones, and you also learned about them in the vital fluid block. Steroid hormones, progesterone, glucocorticoids, mineralocorticoids, female and male steroid hormones, all of them are synthesized from cholesterol. Now, in plants, the plant sterols, including ergosterol, which is precursor of vitamin D in our body, is also put together from cholesterol or synthesized from cholesterol. You will learn in the next block, which is the muscle and movement block, how important vitamin D is in calcium and phosphorus metabolism and in our bone health, of course. Now, let's summarize this, what you learned until now. So we get cholesterol from diet, and it is packaged in chylomicrons. Cholesterols are also de novo synthesized in the liver and packaged in VLDL. And HDL is the one that picks up cholesterol from extrahepatic tissues. So chylomicron, VLDL, this is VLDL, and HDL, all of them are carrying cholesterol to the liver. Now, what it means that the liver is a major decision-making body, again, of what the cholesterol will be used for. So what will liver do? Liver will do packaging of cholesterol, either those that are coming from the periphery or de novo synthesized in the liver, and secrete that cholesterol together with triglycerides in the form of VLDL for the peripheral cells to share. Cholesterol, however, when a lot of them are coming back to the liver, needs to be eliminated. As I said before, the cholesterol ring cannot be opened up, so we cannot catabolize cholesterol. So what do we do with that? We convert part of cholesterol to bile, and then we take free cholesterol or cholesterol ester, and we package in bile. And then through the gallbladder, this bile and the packaged cholesterol will go to the GI. Cholesterol will be released. Bile acids and salts will aid the digestive process. This is a pretty smart way of regulating cholesterol in our circulation and also making sure that the extra can be secreted, can be converted and secreted so that it will not be deposited in our vasculature and will not cause problems, including myocardial infarction, if you recall. So how do we synthesize cholesterol? This is a liver-specific process, although literature says that cholesterol can be potentially synthesized in the sites where steroid hormones are synthesized. But it was shown that majority of the steroid hormones are actually synthesized from cholesterol, which is taken up by these tissues and organs. What is the source? Acetyl coenzyme A. You remember it is coming from glucose. What do we need for cholesterol synthesis? We need ATP, energy, of course, and we need NADPH. You remember that every reductive synthetic step, we need NADPH, and NADPH is coming from hexose monophosphate shunt. So, when glucose is pouring into the liver, glucose will be used to produce acetyl coenzyme A. Glucose will be used to produce ATP in the mitochondria, also acetyl coenzyme A in the mitochondria, and glucose is also be used to produce NADPH in the hexose monophosphate shunt in the cytosol. Where is cholesterol synthesis occur? It actually occurs in the cytosol of the liver. Now, let's see a couple of steps. Again, you don't need to remember the structures, but please remember important steps and, of course, the regulated enzyme, which I will emphasize for you here. So, acetyl coenzyme A, produced in the mitochondria, comes out to the cytosol in the form of citrate, and then it becomes acetyl coenzyme again. Two of them will produce acetyl coenzyme A, and then other acetyl coenzyme A. So three of them will produce hydroxymethyl glutaryl coenzyme A, or HMG coenzyme A in short. The enzyme is HMG-CoA synthase. Now, here it is what you must remember. What happens to HMG coenzyme A and how it goes further for cholesterol synthesis? It is regulated by the next enzyme, which we call HMG coenzyme A reductase. So HMG coenzyme A, using NADPH, you remember hexose monophosphate shunt supplies that, will be reduced, you see, reduced to so-called mevalonate. This is the rate-limiting and heavily regulated step of cholesterol synthesis. All right, let's go further. I put this slide here, and you will see that I will put it in an upcoming lecture too, that the steps are very similar in ketone body synthesis and cholesterol synthesis. Both of them are being done in the liver. However, when two acetyl coenzymes A's are merged and the third is producing HMG coenzyme A, you see this is the same enzyme. HMG coenzyme A will be cut, lysed, and an acetyl coenzyme A will be liberated. And this is the way how we produce ketone bodies. You remember what happened to HMG coenzyme A in the cytosol? So this is in the mitochondria. In the cytosol, it was used to produce mevalonate and produce cholesterol. Now, another thing to remember, that even if these are both liver processes, one is happening in the mitochondria, cholesterol synthesis in the cytosol. Ketone body synthesis is upregulated by glucagon, and cholesterol synthesis is upregulated by insulin. So let me show you this. Again, I will show you this figure again when we talk about ketone body synthesis. So in cholesterol synthesis, HMG-CoA is formed in the cytosol. It comes from glucose. Pyruvate goes to the mitochondria. Acetyl coenzyme A is formed, going out to the cytosol. HMG coenzyme A is formed, and HMG coenzyme A reductase will produce and further steps will produce cholesterol. Insulin that upregulates glycolysis, production of acetyl coenzyme A and energy in the mitochondria, will also upregulate the key enzyme HMG-CoA reductase in the cytosol, so a cholesterol will be synthesized. However, when ketone bodies are produced by the signal of glucagon, then fatty acids and amino acids that are coming into the liver, not glucose, you remember, glucagon signal, liver is not running glycolysis, but takes up fatty acids, takes up amino acids. Amino acids mainly will be used for gluconeogenesis. Fatty acids will be used to produce energy, driving gluconeogenesis included, and fatty acids also will be used to produce ketone bodies. So HMG coenzyme lyase will be the key enzyme, and please remember insulin, cholesterol, glucagon, ketone bodies. So what is happening then later? So here is mevalonic acid, and mevalonic acid, which is a 6-carbon lipid-based molecule, will be decarboxylated. So we have 5 carbon unit intermediates, which will condense with each other until we reach C30, 30 carbon containing molecule, which then will form the first cholesterol ring-based molecule, which we call lanosterol. And lanosterol will be further modified to produce cholesterol. I put here this figure for you, not to be memorized, please. But I wanted to show you that we actually need a lot of energy to drive this process. We also need, of course, basic molecule glucose that will be converted to acetyl coenzyme A forming mevalonic acid, but we also need glucose to be used for redox reactions in the form of NADPH. Right. So mevalonic acid, ADP use producing this 5-carbon unit, which we call isopentanol pyrophosphate, and then this will be fused and fused and fused. You see C5, C10, C15, etc., C30, which then be modified to lanosterol. All the 4 rings that are part of cholesterol are there. And then it will be further modified to cholesterol. So the purpose is to show you that ATP energy is heavily used, and then for every step from C15 to C30 we'll use NADPH, and NADPH will also be used in that monooxygenase system, which will form the first cholesterol ring-based material. Now, let us switch to what happens to cholesterol in the liver. So we synthesized it, correct? So we have cholesterol in the liver, which is newly synthesized. However, in the same time, cholesterol which is coming from the periphery, either as a remnant from VLDL, which was the product of the liver, or it comes through HDL or chylomicron remnants, which I put here, ApoE receptor. It is recognized by the liver by specific receptors, LDL receptor, ApoE receptor, a scavenger receptor for HDL, if you remember, and they will be internalized. When they are internalized, they will go to the endosome and the lysosome. The receptors will be recycled, and everything else will be degraded into their components. So after the lysosomal process, we will get amino acids, fatty acids, and cholesterol. So we have the cholesterol either newly synthesized or mixed with newly synthesized and returned to the liver. And here is the step where the liver should make a decision depending on the intracellular concentration of cholesterol to what to do with that. So cholesterol will be used for cell membrane repair, of course. You remember intracellular, extracellular, cell membrane and intracellular membranes will be repaired. Can be used for steroid hormone synthesis, and a huge amount will be used for bile acid synthesis. Cholesterol, if the concentration is high, partially can be stored for a short time in a form of cholesterol ester as droplets in the liver. Now, what does, however, this intracellular cholesterol signal for? It will signal for reducing the de novo synthesis of cholesterol if cholesterol is coming from the periphery. Very smart. It is not producing cholesterol. Liver is not producing cholesterol if there is enough coming back from the extrahepatic tissues to the liver. This increased cholesterol level will also signal to the DNA through different transcription factors of not synthesizing LDL receptors that would go to the surface to take more LDL. And actually, although it is not labeled here, the oversupply of cholesterol will also inhibit the recycling of the LDL receptor, actually keeping them and driving them to the lysosome for degradation. This is such a wonderful system. It usually works well, except if we humans do something which kind of takes this wonderful equilibrium out of balance. Now, it also comes by genetic predisposition over producing cholesterol, this feedback of cholesterol synthesis not working perfectly, also eating too much cholesterol containing food or too much sugar. Now, I just put here something for you that 1300 milligram of cholesterol is returned to the liver daily. Much of them is coming from lipoproteins, but also bile is also recycled. So this wonderful system of turning cholesterol to bile is there, and it is very important. But because bile is recycled to the liver, not a lot of cholesterol will be used to produce bile. You will see a little bit later of that. All right, so let's see how is this feedback regulation of cholesterol to the key enzyme, HMG coenzyme A reductase, is working. You would think that it is just an easy binding to the enzyme and turning the activity down, feedback inhibition. Well, not such luck. You have a lot to remember. So let's see. So this is the liver cell, hepatocyte. Here is the key enzyme of cholesterol synthesis, HMG coenzyme A reductase. You remember it takes HMG coenzyme A, produces mevalonic acid, which will end up in produce cholesterol. Now, how do we regulate this enzyme? Let's start with the hormones. Insulin upregulates the gene expression of the enzyme itself, HMG coenzyme A reductase, and it also dephosphorylates and activates the enzyme. You remember that insulin signals, and in the signaling pathways, there are phosphatases that are produced. So one of these phosphatases will be dephosphorylating the enzyme and making it active. When glucagon signals to the liver, glucagon downregulates the gene expression, so less enzyme will be synthesized, and also phosphorylates the enzyme and inactivates the enzyme. Now, this is pretty logical, is it not? And it would be enough to regulate the cholesterol synthetic process, but actually our liver is playing safe, and there are additional regulatory steps included. One is the increased amount of intracellular cholesterol is actually not going back and inhibiting the enzyme itself, but actually upregulating the protein kinase, which will phosphorylate the enzyme without hormonal action and inactivates the enzyme. Smart, isn't it? Now, let's go further. Cholesterol itself upregulates its own esterification by upregulating cholesterol esterase, so cholesterol can be put into storage. Cholesterol ester inhibits the replenishment of LDL receptor, if you remember from the previous slide. So it actually signals back to the cell not to take more cholesterol in until the cholesterol can be handled by the liver. Now, related to production of cholesterol and what cholesterol has, so upregulating kinase, inactivating the key enzyme for synthesis, cholesterol also is inhibiting the production of the so-called sterol regulatory element binding protein, which would go to the to the nucleus and would upregulate the transcription of this enzyme, transcription of the gene of the enzyme. And cholesterol also decreases the stability of the messenger RNA of the enzyme. So when cholesterol is synthesized, it is sensitive, the liver is sensitive to the concentration of cholesterol. Cholesterol inhibits its own production through phosphorylating and inactivating the key enzyme, turns down the transcription of the enzyme itself, so it reduces the number of enzymes in the cytosol, and also reduces the stability of messenger RNA, again reducing the amount of the key enzyme for cholesterol synthesis in the cytosol. Now, this is something that I will hold you responsible for, so please remember this particular slide and the words around it. If the natural regulatory system for some reason is not working well, there are drugs which we call statins that can also inhibit the key enzyme. You will hear a lot about these drugs in the pharmacology lectures, but let me just show you that here is the enzyme HMG-CoA reductase, and these statins are actually resembling of the substrate and they bind to the substrate binding site of the enzyme and competitively inhibit the enzyme. Reminder, competitive inhibitors are substrate analogues that bind to the substrate binding site of the enzyme and inhibit the enzyme temporarily. This is not a covalent modification. This is actually binding and releasing the enzyme. And because this is competitive inhibitor substrate analog, when the concentration of the substrate increases, it will compete off the inhibitor. Now, because of that, this category of drugs need titrating. So how much drug, how much inhibitor should be there to, when there is a need for cholesterol synthesis, the substrate, incoming substrate could compete off this inhibitor and cholesterol synthesis would go on. So sometimes these drugs are not enough and they need to be used in combination with low cholesterol diet. So lesser chylomicron remnant or lesser cholesterol would come to the liver by chylomicron remnants. And also there are drugs that are targeting bile salts. So they bind away bile in the GI tract and they would not let bile to be recycled to the liver. Thus, more cholesterol would turn into bile and more cholesterol would be removed from the liver. Now, this is just for fun. So this is out of Lippincott and there is a tiny little bit of mistake here. So they say that this is HMG coenzyme A, the substrate of the enzyme, which is bound to the carrier and forms the inhibitor, which then will bind and binds to the active site of the enzyme. However, if you look here, this is actually mevalonate. Now, this is absolutely true for these inhibitors, that the substrate binding site can either bind the substrate or it also binds the product. And if the product is in high concentration, it will inhibit the enzyme to take its substrate. Now, this is just for fun, for biochemistry. All right. So we synthesized the cholesterol, we regulated cholesterol synthesis. Let's see how we eliminate cholesterol from the liver. Elimination of the cholesterol cannot be done in any other way but in the synthesis of bile acids and bile salts, since we cannot open the ring and we cannot utilize the components of the cholesterol ring to produce energy. So cholesterol is eliminated by converting cholesterol to bile acids and salt, and also these bile acids, bile salts that are produced in the liver will take cholesterol with them and thus reduce the intracellular cholesterol concentration in the liver cells. What are these bile acids and bile salts? If you look at the structure, you see that the cholesterol ring system is still there, but there are modifications in the substituents and also modification of the side chain. Which side chain was an aliphatic side chain in the cholesterol, now it carries a carboxyl group. Bile acids are produced in the peroxisomes of hepatocytes. You may recall that these are CYPs, cytochrome P450 systems that can produce hydroxyl and carboxyl groups. The most abundant bile salt, which is bile acid, which is synthesized, is cholic acid, and it is a derivative of cholanic acid. So again, the first bile salt is cholanic acid, which actually will be turned into cholic acid. So this is the first proper bile acid. You see this carboxyl group here, hydroxyl groups here and there, all produced by CYPs in the peroxisome. Now it is further turned into deoxycholic acid, chenodeoxycholic acid, depending on which hydroxyl of the two are still there, or lithocholic acid when those two hydroxyl are both removed. Doesn't really matter. All of them has the detergent-like activity. They are all amphipathic. However, these are the bile acids, and many of the bile acids are not acting in the free acid form, but these carboxyl groups are esterified, conjugated with either glycine or taurine, which is also an amino acid derivative, and they are bound to this carboxyl group, esterifying that, and then the polarity is enhanced. So amphipathic character, which was carboxyl group, now we have extra hydrophilic groups produced by the conjugation of the bile acid to form bile salts. Now we are talking about primary and secondary bile acids. Primary bile acids that are produced in the liver and secondary bile acids that are produced in the intestinal tract by the microbiota, by microorganisms that are there in our GI tract. Just a couple of words about cholic acid synthesis. The only thing that you need to remember, although if you like biochemistry and you want to see what is happening to cholesterol to form cholic acid, it is worded out here. The only thing that I want you to remember that it is cholesterol 7 hydroxylase, which actually is a CYP. So every modification of the carboxyl group formation, extra hydroxyl group, is done by cytochrome P450 system. Now, this enzyme is also regulated when there is a need for bile acid synthesis. Cholic acid is going back and inhibiting its own synthesis. When cholesterol amount or concentration is increased in the liver, then it activates the enzyme and turns cholesterol to cholic acid. Why do we produce bile acids and bile salts? We produce them because cholesterol concentration is decreased, and also cholesterol can be excreted with the help of bile acid, So cholesterol level is decreased, as I said, in the liver. Bile acids, together with phospholipids, as I put here, solubilizes cholesterol in the bile. So prevent precipitation of cholesterol in the bile, in the gallbladder. So it actually helps to prevent gallstone disease. If there is not enough bile acid produced or there is no phosphatidylcholine produced by the liver, then cholesterol can precipitate, causing gallbladder problems, gallbladder stones or gallstones. And you remember in the GI tract, bile acids and also phospholipids emulsify the dietary lipids, triglycerides, dietary cholesterol esters, and let the pancreatic enzyme to bind to this micelle and start digesting lipids to be able to absorb by the mucosal cells. Bile acid activates also the lipases, and bile acids facilitate the absorption of fat-soluble vitamins, including vitamin D, K, E, and A from the intestine to the mucosal cells. Now, let us talk about what I mentioned before. We do produce bile acids and bile salts, and we also recycle them. So here is the primary synthetic pathway. Cholesterol is synthesized in the liver, turns into primary bile acids. They go to the bile duct, gallbladder. By the signal of cholecystokinin, they are released to the GI tract. Primary salts have primary bile acids and salts, their function, and then they are turned into secondary bile acids and salts. Some of them will be excreted after their function is done, but the majority of them will be recycled to the liver. So if they are recycled, primary and secondary bile acids, together with glycine and taurine, which was modifying the bile acids to bile salt, all goes back to the liver. They will be primary, secondary, they will be part of the pool of primary and secondary bile acids and bile salts. They go again to the bile duct and the gallbladder, and they go to fulfill their function in the GI tract again. This is actually a very nice system because you remember it is very expensive to synthesize cholesterol. It is even more expensive to produce primary bile acids. So this recycled enterohepatic circulation of the bile saves a lot of energy for the liver. So look at the numbers. Bile is, as a primary, produced 15 to 30 grams per day. It goes out to the circulation, and only 0.5 gram of this is excreted. So mainly almost 15 to 30 gram is recycled to the liver. So de novo only 0.5 grams will be produced daily. Very nice system. However, as every very nice system, there can be problems with that, which result in certain pathologies. So let's have a look at that. What can go wrong is gallbladder can be full with cholesterol precipitates because something is wrong with the bile acid salt synthetic or recycling system. What can cause bile salt deficiency so that cholesterol can precipitate in the gallbladder, forming gallstones? So it could be malabsorption of bile acids, the recycling of bile acids from the intestine, and suddenly the liver needs to synthesize much more, And until it replenishes the necessary amount of bile acids and salts, cholesterol can go out and precipitate in the gallbladder. It can also be a problem with obstruction of the biliary tract, which means that everything will be bagged up. Cholesterol can precipitate, but if it is bagged up, the recycling will not happen and there will not be enough bile acids and salts in the GI tract to fulfill their function, so there will be digestive problems. Severe hepatic dysfunction, which means that there will not be enough cholesterol taken up or synthesized, not enough primary bile acids produced, not enough to eliminate the opportunity for cholesterol precipitation, and neither enough to fulfill the GI function. That actually is a huge problem in liver disease, low bile acid and gallstone formation, and digestive problems will occur. However, there could be other problems too. For example, excessive feedback suppression of bile acid synthesis. So it is excessive feedback that there will be not enough produced, and the same thing happens. A problem with cholesterol, problem with GI tract function. An increased bile acid excretion, which will take a toll on the liver to synthesize more, and again, same problems can happen. So some of the treatment options are actually laparoscopic surgery. So they help to take out gallbladder if there are stone formation there, and also administering secondary bile salts to the digestive tract to have enough to aid digestion. Now, this is again just a reminder. You already learned about this in the vital fluids block, that cholesterol is the sole precursor of the synthesis of every class of steroid hormones. You may recall that the first enzyme, which is actually a CYP and catalyzes 3 steps between cholesterol to pregnenolone, is called desmolase. And this is a mitochondrial P450 or CYP. It also uses oxygen, uses NADPH as of typical of CYP systems. And then every other step in the target organs, let it be adrenal gland to glucocorticoid and mineralocorticoid, or the sex hormones, sex organs, ovaries and testes to produce the sex hormones. Every other modification is a CYP enzyme. Now, this is a great summary figure with some of the regulatory steps included for cholesterol synthesis, and a little reminder for you of lipoproteins. So what did you learn today? Cholesterol, which has a complex ring structure, has many physiological functions too. So to be able to synthesize cholesterol is an important biological function of the liver. Cholesterol derives from food and also de novo synthesis in the liver. Cholesterol and cholesterol esters, either from the liver or from the food source, delivered to the other cells through lipoprotein delivery. Please remember chylomicrons and VLDL, which goes back to in the remnant forms to the liver, chylomicroremnant, LDL, and the HDL, which sole function is to pick up cholesterol from the periphery. Receptor-mediated endocytosis, which also takes care of determining how much cholesterol is in the liver. Cholesterol is synthesized from glucose, acetyl coenzyme A, And glucose also supplies the ATP and NADPH, which is necessary for the synthetic process. HMG coenzyme A reductase is the rate limiting and key enzyme, heavily regulated. Please remember all the regulatory steps that are involved in regulating this key enzyme and cholesterol to reduce its own synthesis. Please remember statins are competitive inhibitors of the same enzyme. Cholesterol also is a starting material of synthesis of bile acids and salts. It fulfills this function of eliminating extra cholesterol from the liver, but also putting bile acid through the gallbladder to the intestines aids the digestive process. Please remember the enterohepatic circulation of the bile acids back to the liver. And please remember that cholesterol is the sole source of synthesizing steroid hormones. Now, this was a substantial lecture. And again, if you want to read the content of this lecture, you will find the Word document in that special folder on Entrada. Thank you for your attention.